Let's go over the Chapter 11 Study Guide, Section 1, Basic Patterns of Human Inheritance. You can refer to the 11.1 Active Reading Review. You can refer to the notes for all of these. So let's go ahead and get started. A pedigree shows the inheritance of a particular trait over several generations. An organism with two of the same alleles for a particular trait is said to be homozygous for that trait. Now that can be homozygous dominant or homozygous recessive. An organism with two different alleles for a particular trait is heterozygous for that trait. When alleles are present in the heterozygous state, the dominant trait will be observable. The phenotype will be the dominant phenotype. An individual who is heterozygous for a recessive disorder is called a carrier. Examples of recessive genetic disorders in humans are cystic fibrosis and albinism. Those are the two examples we referred to in the notes. And obviously 8 and 9 can be switched. They don't have to be in that specific order. Okay, next up, we had a table of different genetic disorders. So let's go ahead and start with the first one, number 10, caused by altered genes, results in lack of skin pigment. That's albinism. Number 11, recessive genetic disorder characterized by body's inability to tolerate galactose. That's galactosemia. Recessive genetic disorder, gene found on chromosome 15, characterized by lack of enzyme that breaks down fatty acids. That's Tay-Sachs disease. Number 13, recessive genetic disorder affects mucus-producing glands, digestive enzymes, and sweat glands. That's cystic fibrosis. 14, dominant genetic disorder affects the nervous system. That's Huntington's disease. And then the last one, autosomal dominant genetic condition affects height and body size, chondroplasia. All right, a little continuation of section 1. True, false. A scientist uses a pedigree to study family history. That is, in fact, true. A pedigree traces the inheritance of a particular trait through only 2 generations. That's false. It can be many, many, many generations, depending on how much data the scientist or geneticist has. In a pedigree, one who does not express the trait is represented by a darkened square or circle. That's actually false. They would not be darkened or shaded in or colored in. In a pedigree, a horizontal line between 2 symbols shows that these individuals are the parents of the offspring. That's true. And then, of course, you see there number 20 regarding the small pedigree example here. Individual 2-1, as shown below, is in generation 2. Well, yeah, that is true. It says it right here. All right. Refer to the pedigree for these questions. Recall, if the trait is recessive or dominant based on the following information. In a pedigree, individuals 1-1 and 1-2 are unaffected but have an affected child. That would mean that it would be recessive. Number 22. Looks like we have a visitor here. 22. Specify if parents 2-1 and 2-2 who have an affected child are carriers of the trait. They are carriers. So if we look here, it doesn't have... Now, if this were a pedigree with half shaded in symbols, they would be halfway shaded in, representing that they are carriers. The reason we know they're carriers is because they had at least one affected child. Okay. Okay, number 23. Tell whether there is a dominant gene in the genotype of 2-4. There has to be at least one dominant gene. Okay, we look here because they all have these 2, 2-3, 2-4, all had unaffected children. Okay. Okay, moving on. Section 2, this is a quick one, dealing with, in this case, incomplete dominance. Okay, we have R prime as the genotype that represents the sickle cell, red blood cells. R and R prime is both, and RR would be normal red blood cells. Okay. All right. Refer to the Punnett square. Some reminders there. I'm just going to zip right through this. Tell if the father has color blindness. No. As you can see there, he does not. Little b is recessive. Here's the father, XY. He does not have a little b. Therefore, he is not colorblind. Specify if the father has a recessive allele. No, he doesn't. State whether the only child that could have color blindness is male or female. The only child that can have color blindness between these two parents is a male right here, because the father can only give the unaffected X. So therefore, every female that these parents have will be unaffected. However, one of them could be a carrier. All right. And lastly, section 3, micrographic chromosomes. That's a karyotype. Abnormal number of chromosomes caused by non-disjunction. Withdrawal of tissue from the placenta. Chorionic villus sampling. Extra chromosome 21. Down syndrome or trisomy 21. And number 5, protective cap at the end of a chromosome, a telomere. All right. Here's some drawings. I did my best here. So within Down syndrome, chromosome 20, the pair is normal. Chromosome 21, there's 3 of them, hence the nickname trisomy 21. Chromosome 22 has the 2 chromosomes, one from mom, one from dad. Tell why this karyotype is called trisomy. It has 3 chromosomes of one kind. That's within the 21st pair, what should be a pair, obviously not in this case. Recall the term for the sister chromosomes failing to separate during cell division. That's called non-disjunction. And lastly, state whether the risk of having a child with Down syndrome is higher in mothers who are younger or older. The answer to that is there's a higher risk, greater risk in mothers who are older. All right, so that's it for the Chapter 11 study guide. And I'll see if you guys can pause it there, or if you need to pause it there. You're all set. So hope that helps you. I will post the exam, and you will have, I'll post everything on Classroom Directions. All right. Thanks for sticking around this long, and I will see you guys, well, hopefully soon. This is Mr. Case signing out.